Good morning. Today we are going to take the remaining part of our sixth chapter, Unraveling Genetic Histories. Last class or previous classes we have studied about the Mendel's cross, that is about monohybrid cross, dihybrid cross, and all we have studied. And you have seen the videos related to monohybrid cross and dihybrid cross. Now, today we are going to study about the structure of DNA. So we have told that earlier class we have told that River John Mendel, who is the father of genetics, so he told about that a factor is responsible for the transfer of characters from parent to offspring. So he just know about a factor is responsible for that. But what is that factor responsible for the transfer of these characters? He didn't explain about those characters. But later, other scientists studied about that. They found out that genes are the factors responsible for the expression or transfer of these characters from parent to offspring. So these genes, so we have to know where these genes are located. These genes are located on DNA. DNA are present on the chromosomes, and these chromosomes are present in the nucleus. Okay, so you have already in your 8th standard you have studied about the structure of a cell. In the cell, what is present in the center? Nucleus is present. On the nucleus, chromosomes are present. On the chromosome, DNA is present. On the DNA, genes are present. Is it clear for you? So once again, I will repeat it for you. The center of the cell is called as the nucleus. Inside the nucleus, what is present? Chromosomes are present. On the chromosomes, DNA is present. On the DNA, genes are present. Is it clear for you? So DNA, on the DNA, genes are present, and these genes are responsible for the transfer of characters from the parents to offspring. Parents to offspring means what? The next generation. So you may be heard about DNA earlier. So DNA, full form of DNA is deoxyribonucleic acid. So DNA is deoxyribonucleic acid. The sugar present in DNA is deoxyribose sugar, is the sugar present in DNA, and the full form of DNA is deoxyribonucleic acid. So the structure of DNA is found out by two scientists named James Watson and Francis Crick. So Watson and Crick is the scientist who found out the structure of DNA. The scientist James Watson and Francis Crick found out the structure of DNA. DNA is having a double helical structure. Okay, so DNA having a double helical structure. Now we can see the structure of DNA. Here in the picture you can see the double helical structure of DNA. In that, two long strands, you can see two long strands of DNA, and in that you can see that it's labeled as rungs. Rungs are the steps that's seen in that double helical structure, and that steps are nitrogen bases. In the picture you can see 4 nitrogen bases are present for a DNA, that is adenine, thymine, cytosine, guanine. Once more I will repeat it for you. Adenine, thymine, cytosine, guanine are the 4 nitrogen bases present in the DNA. And adenine always pairs with thymine. You can see in the picture, adenine, thymine, every nitrogen bases are given a particular color. So according to that color, you can identify in the DNA. In the picture you can identify. That is adenine and thymine. Adenine and thymine always pairs with the adenine always pairs with the thymine. That you should keep in mind. Adenine always pairs with the thymine. And cytosine and guanine. Cytosine always pairs with the guanine. So DNA is having a double helical structure. In that double helical structure, two long strands, two long ribbon-like structures you can see, and two long ribbon-like structures, and inside those ribbon-like structures you can see rungs, and step-like structures is known as the nitrogen bases. These nitrogen bases are four kinds of nitrogen bases: adenine, thymine, cytosine, and guanine. Adenine always pairs with the thymine, cytosine always pairs with the guanine. And these strands, DNA strands, are made up of, you can see in the picture, phosphate and sugar molecule, nitrogen base. These are the three components of a DNA strand. And this phosphate molecule, sugar molecule, and nitrogen base together, these three together forms the nucleotide. So what is a nucleotide? A nucleotide consists of a sugar molecule, phosphate molecule, and a nitrogen base. So DNA molecule is made up of units called nucleotides. You can see in the picture, if we are taking one strand of DNA, you can see in that picture a sugar molecule, a pentagon shaped sugar molecule is there, and a phosphate molecule is given attached from the side of that sugar molecule, and from the other side of the sugar molecule you can see a nitrogen base is attached. So these three forms a nucleotide. So DNA molecule is made up of units called nucleotides. A nucleotide contains a sugar molecule, a phosphate molecule, and a nitrogen base. And DNA, each strand consists of different, so many number of sugar molecule and nitrogen base and phosphate. DNA has, generally, a DNA has 4 nucleotides. Okay, why? If a question comes, why a DNA consists of 4 nucleotides? Because the answer is because DNA consists of 4 nitrogen bases. I have told you about the 4 different kinds of nitrogen bases. That is adenine, thymine, cytosine, and guanine. These are the 4 nitrogen bases. These 4 nitrogen bases, because of these 4 nitrogen bases, DNA consists of 4 nucleotides. I have told you about the structure of a nucleotide. Each nucleotide consists of a sugar molecule, phosphate, and a nitrogen base. So, as each nucleotide consists of a nitrogen base, there are 4 different kinds of nitrogen bases are present in a DNA. As there are 4 different kinds of nitrogen bases, a nucleotide DNA consists of 4 different kinds of nucleotide. Is it clear for you? Generally, 4 nucleotides are present for a DNA. The reason for that is because of the presence of nitrogen bases, 4 different kinds of nitrogen bases. That's about the DNA. Now we can see about the difference between the structure of DNA and RNA. Here, I have told you the full form of DNA is deoxyribose nucleic acid, and the full form of RNA is ribonucleic acid. Here, a comparison between DNA and RNA is shown. Number of strands in DNA is 2. Double helical structure of DNA you have seen. Number of strands in the DNA is 2. As compared to RNA, RNA is having only single strand. So when compared to RNA, DNA is a double stranded and RNA is a single stranded. Then type of sugar. Deoxyribose sugar is present in DNA. In the case of RNA, ribose sugar is present. Then type of nitrogen bases. DNA consists of adenine, thymine, guanine, and cytosine. And RNA, adenine, uracil, cytosine, guanine. So you should keep in mind a nitrogen base is different in the case of RNA. A single nitrogen base is different in the case of RNA. That is, instead of thymine in DNA, in RNA, uracil is present. So DNA consists of 4 nitrogen bases, that is adenine, thymine, guanine, cytosine. RNA also consists of 4 nitrogen bases, but instead of thymine, uracil is present in RNA. That is adenine, uracil, cytosine, and guanine. So I think it's clear for you about the comparison between DNA and RNA. DNA is deoxyribose nucleic acid. RNA is ribose nucleic acid. DNA is double-stranded. In the case of RNA, it's single-stranded. And in the case of DNA, the nitrogen bases present is, which are the nitrogen bases present in DNA? Adenine, thymine, guanine, and cytosine. And in the case of RNA, instead of thymine, uracil, nitrogen base, uracil is present in the case of RNA. That's about the comparison between DNA and RNA. Now, action of genes. So, in our body, each function is controlled by the genes. Our metabolic activities and all our functions happening in our body is controlled by the genes. Production of certain proteins, everything is controlled by the genes. And where these genes are present, genes are located on the DNA. So now we can see the steps in the production of certain proteins. And we can see that how it's happening, how the genes are acting. So I have already told you that genes are present on the DNA, and this DNA is present on the chromosomes present in the nucleus. So here the structure of nucleus is shown. In that you can see, inside that nucleus you can see a double helical structure, that is DNA. And DNA directly did not participate in the protein synthesis. So that function is done by the genes. The gene gives the message through the RNA. mRNA means messenger RNA from the DNA is coming out. You can see a red ribbon-like structure, that is mRNA, messenger RNA, carrying the message from the gene present in the DNA. What's the message? That is, a protein should be produced inside the cell. So once more I am repeating it for you. DNA did not participate directly in the protein synthesis. So DNA, the genes are present on the DNA. The genes give the message through the RNA. That is that red ribbon-like structure shown in the figure, that is mRNA. mRNA means messenger RNA. This messenger RNA carries the message that a protein should be produced inside the cell. So that messenger RNA carrying that message is coming out through the nuclear pore present on the nucleus. You have studied the structure of nucleus in the 8th standard. So through that nuclear pore, that mRNA is coming out from the nucleus into the cell. When it reaches the mRNA reaches outside the nucleus. You can see that mRNA is reaching outside the nucleus. When it reaches outside the nucleus, you can see the structure ribosome is present. You have studied about the cell organelles, and you have earlier studied about the cell organelles. In that, you have studied that the center of protein synthesis, ribosomes, is the center of protein synthesis. So this mRNA carries this message to the ribosome, cell organelle ribosome. When this mRNA with the message, a protein should be produced, and that with that message, when this mRNA reaches the ribosome, tRNA, transfer RNA, brings different amino acids, different kinds of amino acids to ribosome. So different kinds of amino acids are scattered like bead-like structures. You can see in the cytoplasm, bead-like structures are present. Those bead-like structures are different kinds of amino acids. Those amino acids are taken to the ribosome with the tRNA, transfer RNA. Why amino acids are taken to ribosome? Because amino acid is the single unit of protein. So the single unit of protein is the amino acids. Amino acids join together to form the protein. So these different kinds of amino acids are taken to the ribosome, and when it reaches the ribosome, a protein is produced and this protein is produced with the help of mRNA. mRNA is taking the message to ribosome, and this message reaches the ribosome with the help of tRNA. tRNA carries these amino acids to the ribosome, and a protein is made with the help of transfer RNA and ribosome. In the ribosome, this message is reached, and in the ribosome, protein is synthesized. Is it clear for you? And another kind of RNA present is rRNA, ribosomal RNA. It also helps in the protein synthesis. Is it clear for you? So these different kinds of, with the help of these different kinds of RNA, what happens inside the ribosome, a protein, a new protein is synthesized. Why tRNA is collecting different kinds of amino acid to the ribosome? Because it's because amino acid is the single unit of protein. So join these amino acids, what happens? We can form the proteins. Is it clear for you? So this is the action of gene. That's about the action of gene. Gene cannot directly participate, not gene, DNA cannot directly participate in the protein synthesis. With the help of the gene only, the gene is giving the message a protein should be synthesized through the mRNA. mRNA means messenger RNA, and this messenger RNA is taking the message to the ribosome. Why this messenger RNA is taking the message to ribosome, the cell organelle ribosome? Why can't it take to mitochondria? Because in the cell organelle, ribosome is known as the center of protein synthesis, and mitochondria function is what? It helps in the production of energy. So it's entirely different. Each cell organelle is having a specific function. So the message that is a protein should be synthesized, that message is given to the ribosome, and this mRNA, when it reaches the ribosome, with the help of tRNA, transfer RNA, collect the amino acids, different kinds of amino acids. They collect the amino acids and collect and transfer it to the ribosome. And when these different kinds of amino acids reaches the ribosome, a protein is synthesized there. Okay. So that's about the protein synthesis. The different kinds of RNA taking part in protein synthesis is rRNA, that is ribosomal RNA. Ribosomal RNA will be seen attached to the ribosome itself, and mRNA, messenger RNA, that is the RNA that is carrying the message from DNA to DNA to the ribosome. Then tRNA, that is transfer RNA, that collect the different kinds of amino acid and transfer it to the ribosome. And with the help of this RNA, what is happening? The action of the gene, that is the gene is the message given by the gene. A protein should be synthesized inside the cell. A protein is synthesized in the ribosome. With the help of this RNA, a protein is synthesized in the ribosome. Okay, then next is about the chromosomes in human. There are the total number of chromosomes present in a cell of human being is 46. Okay, so in this 46 chromosomes, 44 chromosomes are somatic chromosomes, and 2 chromosomes are sex chromosomes. So the total number is 44 plus 2. And this 44 plus 2, 44 is somatic chromosomes. Somatic chromosomes means doing the other functions. And those chromosomes, that is 2 chromosomes are present, that is sex chromosomes. The sex chromosomes control the sexual characteristics. In female and male, the sexual characteristics are controlled by the sex chromosomes. There are 2 kinds of sex chromosomes, that is X and Y chromosomes. X and Y chromosomes are the 2 sex chromosomes present in the human beings. In the case of females, the chromosome number is 44 plus XX. So the genetic constitution of female is 44 plus XX. You should keep in mind that I'm telling about a single cell, not about the full human body. Inside a single cell, in our body, millions and millions of cells are present. But I'm telling about only a single cell. In a single cell, 44 plus 2 sex chromosomes, that is total 46 chromosomes are present inside the nucleus of a single cell. In that, 44 are somatic chromosomes and 2 chromosomes are sex chromosomes. The 2 kinds of sex chromosomes are X and Y. And the genetic constitution of female is 44 plus XX. And the genetic constitution of male is 44 plus XY. Is it clear for you? So the genetic constitution of female is 44 plus XX and the genetic constitution of male is 44 plus XY. I think it's clear for you. And the remaining portion we can study in the next class. Okay. So if you are having any doubt regarding this class, we can clear it now itself. Okay. Thank you.